Thank you so much for the introduction and also for the invitation. I'm very excited to be here today. I think you have a very important mission to educate the next generations in science and technology, and I like to contribute somehow. And in particular, kind of personally, I was inspired by my science teacher when I was 14. It was the first physicist I met, and she made me at the end go into science, and very grateful for that. So, and today, all the talk is going to be around mitochondria and all the spatial models and things that we develop related to mitochondria. But in general, it's about spatial modeling and tools that we and others are developing to doing this. And this, as I mentioned, this project was part of my PhD, the beginning, that I also did in collaboration with the lab of Dr. Sejnowski here at Salk. And mitochondria, as you might know, is really at the core of cell functioning, producing ATP, buffering calcium, regulating apoptosis. In particular, their role is very important at presynaptic terminals that are far away from the soma, and mitochondria is very scarce, are perhaps 2, 3 at most, and at presynaptic terminals. This is a place where neurons communicate, right? I mean, we have a presynaptic terminal, postsynaptic, and we are focusing on mitochondria at the presynaptic terminal. And here I show you some of the processes that require ATP to maintain the transmission. And calcium, once an action potential arrives and calciums come in, then the calcium level needs to go to vessel levels, and for that you need energy to take the calcium out, and many other processes. And for long, it has been shown that the morphology of mitochondria is related to function, in particular focusing on ATP production. And this is work from Hackenbrock from the early 60s. Here I'm showing you electron microscopy images of mitochondria. And as I mentioned, mitochondria has 2 membranes, and here you barely in this one can see both membranes. This is a cross-section of a mitochondrion. And the internal membrane has the number of invaginations that I'm going to show 3D reconstruction where you really can see that in more detail. But here, what he found is that when mitochondria is in a resting state, it's not producing ATP. You see, as the image on the left, you see this kind of images. But when it's in an active process of producing ATP, the morphology is very different. And in other people, more recently, has been shown something similar in fast spiking interneurons. So these neurons actually fire, as its name it says, very fast. So more energy is needed at those synapses to maintain the transmission. So the question that we wanted to address in my project was to see if mitochondrial morphology affects somehow ATP production. And to do that, we decided, to gather a team of interdisciplinary researchers, and what we did was to develop a multi-scale model that has different components. I mean, one and the most, I think, important is that we generated 3D geometries from serial electron tomography. And this is just one of the examples. In particular, for this publications, we only had one. And we combined that with, so in our model, we have a number of proteins, and we need to set kind of the rules of how these proteins are going to behave and the dynamic change. So these are, these are what I'm showing you here, kind of the rules. And then some of these molecules can move in space, so we also add random walk diffusion. And we are integrating all this in a program that is called MCell that has been developed here at Salk and the lab of Dr. Sejnowski, long time ago, but we keep developing things and adding new features. And so we integrated all this with this software. It's actually it's an agent-based reaction diffusion simulator that I will introduce later today in the lab rotation. And so in this case, what we could do is to explore the spatial organization of the main components and also how the components interact with the specialized geometry. And in this first publication that we only had one reconstruction where we did to understand the impact of the morphology was also to develop in parallel a model that was a space independent. I mean, we used ordinary differential equations. So we assumed that mitochondria was just a point. So to have something to compare. And the main conclusion, very, very summarizing, is that the morphology what it's doing is actually buffering buffering ATP. So ATP, you cross the two membranes in mitochondria, and the inside is called the matrix, and that's the place where ATP is produced. And ATP needs to go out. So it's in that going out that the transporters, part of them are located at the rim of the cristae. And so the ATP goes out and gets trapped in those cristae and then goes out. So this was one of the main conclusions from this part of my research. But then the question was, how different is the mitochondrial morphology at synapses? I mean, we only had one reconstruction. So we went and we reconstructed 12 other mitochondria. And I should say these are all from the cerebellum, from axonal and synaptic mitochondria. And here I'm showing you, these are three membranes, but actually there are only two. So the red one is the exterior one, and the other two are so this one is the inner membrane, but you can't see all the internal structure. So I put it, next to this one, but it's actually inside. And so we observe a lot of diversity. So what we decided to do was to use new tools from differential geometry to characterize the morphology. And we compute something that is called the curvature, the first principal curvature, and we were able to distinguish between these two groups that we call global and elongated using machine learning techniques. And then we relate those features of the external membrane of mitochondria with internal ones. And here I'm showing you two examples of how the inner membrane or the cristae membrane actually inside mitochondria looks like for two of these representative example from the group. And you see that they have way more, this openings are called cristae junctions. The globular group has way more cristae junctions and many other features. Another thing that we measure were the areas of high curvature in the cristae membrane. And here I'm showing you that in colors, and in particular we use the threshold, and we are only looking at where the curvature is higher than 80 micrometers to -1, and all the other places we put it in gray. And this is very important, and we quantify these areas of high curvature. And this is very important because it's the place where actually ATP synthase accumulate. And ATP synthase are the enzymes that produce ATP. So we consider that these areas are a proxy of the energetic capabilities of each of these organelles. And I want to mention that we had the help of many high school students for this project. I mean, first, Emily Liu joined us, and she, helped us a lot to get this first reconstruction. and then other students, Rachel Mandelson too, help us to get more. We work very hard to have really high quality measures because we need that to do computational work, and we need that to use these tools that I was mentioning to quantify the curvatures. And both Emily's is now at Stanford, and Rachel is still Salk, but planning to leave soon. So again, I mean, we now have all this structural information, and what we want is to combine this with mathematical modeling. And so we took a similar approach, but now combining the 3D reconstructions that we have with the model that we developed with the different components, particularly the main components, I didn't mention that, but it's ATP synthase and ANTs that are the proteins that translocate ATP. But now what we have is these new tools that quantify the curvature. So how to incorporate that into the models. And the way that we did it is these areas of high curvature allow us to estimate how many ATP synthases are in each of these reconstructions. So we can use and where they are. So we can use all that information, as I'm showing here in this reconstruction, to locate ATP syntheses in these regions. And then go and perform the spatial simulations and get quantitative measures. I mean, here I'm showing as an example the number of ATP in the cytosol, for example, in different configurations. So we did this for all the reconstructions that we have that I show you. And here is, for the globular set and the elongated, and here is where, for one run, we allocated ATP synthases based on the areas of high curvature, and the same for the elongated group. And we quantify the ATP production rate, that is, I'm showing you here, and we observe that actually the globular set produce more ATP, or the ATP production rate is higher than the elongated set. And this project was done in collaboration with another high school student, Kavya Gupta, that, she helped us to set up the simulations. I mean, we had to do this for many, well, as you see, for different reconstructions, and this was demanding, and Kavya helped us a lot with that. Kavya is also now gone. She's doing her undergrad at Yale, and she might be back at Salk for the summer, but we still are talking about that. Let me summarize. I think I rushed a little bit, but I hope that we will have more time for questions. But so we generated 9 reconstructions, and I want to emphasize again that these are really high quality reconstructions. We did the effort because we need to have them to run the simulations. We used a number of new tools from differential geometry that were not used before to quantify and characterize the morphology. And we combine all that morphological information with the mathematical models. And some of the predictions of the models are the ATP's buffer at the cristae due to the morphology. And also, one thing that I didn't show you today, but was a conclusion from one of the publications, is we try to understand if the projection or the export of ATP is limiting the amount of ATP in the exterior, and it's actually the production what limits the amount of ATP outside. And lastly, the rate of ATP production is larger in these globular organelles. Some ideas for the future. I mean, This project was actually motivated by the dysfunction of mitochondrial and Parkinson's disease. I did my PhD in an institute, an assistant biomedicine institute that had a focus on Parkinson's. We didn't come back to those original ideas, but there are some projects and now that we hope to get there. I mean, the idea is basically to use fibroblasts from PD patients and, derive from the fibroblast neurons and use mitochondria from those neurons. And, we want to see if there are structural differences in these organelles too. One of the things that also is a little bit missing that I wish we can do in the future is to validate more the predictions that we are doing with more experiments. And we have collaborators at UCSD developing ATP sensors. And the idea and the challenge is that, to validate our results, you will need to really measure the ATP production or the ATP concentration for one specific organelle, and we should also be able to measure something else, either the volume or ideally to have a reconstruction, but I think we are a little bit far from there. And then one thing that I also mentioned is related to the ultrastructural mitochondria at presynaptic terminals, the work from Threap et al. And that this is also associated with how strong the synapses are. And my work now is more focused on synapses nowadays, and I'm thinking a lot about the strength of the synapses. But perhaps we will come back to this idea that the structure of mitochondria can tell us something about how strong synapses are. And so one thing, Blender, I don't know if some of you might know it, is an open source 3D computer graphics software that is a lot of people is using it, and it's very good to do animations, visual effects, and video games. We are using the software as I put it as a central hub for spatial modeling, because we are adding tools that we need to this software, but we are using Blender as the main kind of a structure. It has many advantages, and that's why we are doing it. But and one of the things why it's so good is that it's extensible. You can manipulate the meshes using Python, and whatever you need to do, you can do it. I mean, just writing code and creating kind of your own add-ons. In particular, today, if you come to a rotation, I mean, we will see this is one of the main stars from all the tools that we develop that is focusing on a spatial modeling. And this is what I mentioned before. EMSA has an add-on in Blender that is called Salk Blender, where you can run your simulation and set up your simulation through this visual interface. You can do everything actually from this visual interface. And I think it's very, it might be, for kids attractive. I think, anything that has the word video games on it could be something to attract them. So I just wanted to mention this. And we will talk way more about Blender in the rotation. And I'd like to thank all the people that work in this project. And so all the experimental data, the serial tomography that we are using is coming from Mark Ellisman lab. Yeah, that's it. So thank you very much for the attention.
We have time for probably about 5 questions.
Yeah.
Is there a genetic tendency for globular or elongated mitochondria?
Not that we know. It might be related to the stage of a particular synapse. If, one synapse is more active, in those cases, mitochondria might be more globular. But we need to answer your question precisely, we will need to do more reconstructions and also measure in another way the activity of the synapse.
Is that one of that, or if it were age-related, especially with Parkinson's?
Aging, yeah, it's absolutely something, and we have here Courtney Glavis. We have a staff scientist here at Salk looking at also mitochondria in aging in macaques. I can't remember all the details, sorry, but yes, it might be related to aging, and that's why we want to continue also with the Parkinson's disease.
So when you have the two mitochondria, is surface area an issue because they seem to be different in shape?
Yeah, we quantify everything. I mean, surface area, volume, and a lot of things derive from those quantities. But no, the volume in the two groups was not significantly different. No.
How do you harvest the mitochondria?
Well, I haven't, I don't do experimental work. So that is, all the guys on Mark Ellisman lab were the ones that did it. But this, I mean, I don't harvest mitochondria. This was, they take a piece of tissue and they use it for electron microscopy. So you see, and indeed, I mean, if you come to a rotation today, I mean, I was planning to start showing you the tomography, so you have an idea of how we start the process, and yeah, so you will see we have many synapses together, so it's kind of a piece of a brain. Yeah.
What's your process for selecting and onboarding the students that you work with?
Yeah, that's always it's hard. I mean, I honestly didn't take the decision with any high school student. I mean, I had to decide with undergrads, but not with high school students. And I was lucky because Kavya, for example, she was working in the lab already. In 2021, she did a summer rotation, and in 2022, I asked, I need help, and talking with another postdoc, she mentioned, Kavya could help you, and that's how things started. But usually they get to us. I mean, it's not that we want to find high school students. I mean, so they are somehow kind of motivated to get into science. And
Were some of those students in high school, or?
the students were high school students. Yes. Kavya was high school student, and indeed, I mean, she did. That's why I was saying. I mean, she worked with us first during the summer only. And I think it was in 22 that we continue a little bit during the year, but that is harder for them. But Any other question? Yeah.
How many powerhouse of the cell jokes do you get?
How many what?
My students, when I'm in your mitochondria, they say powerhouse of the cell.
Yes.
Do you get a lot of jokes about that or no?
I think people also get a little bit, I don't know. No, but not many actually. Yeah, not many.
Thank you.
You're welcome.